Part 1. Life on Earth is diverse and fascinating. It contains the smallest of creatures, such as unicellular amoebas, all the way up to complex organisms such as humans, with intricate internal systems and a large capacity for reason. At first glance, you wouldn't think that these organisms have much in common, but it turns out that they do. Every single living organism on Earth is made up of the same genetic code, the same 4 nitrogen bases that code for every aspect about us. Changes to a genetic code can change the physical characteristics of an organism, and as such are called gene editing. From the earliest forms of unintentional gene editing all the way up to the most recent and advanced form, known as CRISPR, gene editing not only is an incredibly interesting field of science, but one that has the potential to change the future of mankind as we know it. Part 2. The earliest form of gene editing was selective breeding and was mainly used in agriculture. Farmers would take plants or animals with desirable characteristics and breed them, thereby propagating their genes. An example is the selection of wild bananas that had thick skins and large seeds to the bananas that we know nowadays. In 1951, Watson and Crick discovered the double helix model of DNA, and research exploded from there. One of the first experiments involved exposing plants to radiation in order to observe the effect it had on their genes, and observe beneficial, neutral, or harmful changes to the plant, the first induced mutations. The next step was the creation of recombinant DNA, which is a singular piece of DNA taken from multiple organisms. This created genetic sequences that were never before seen and opened up the way for gene therapy, which is the injection of healthy genes into diseased patients to try to cure diseases such as AIDS. In 1974, the first genetically modified animal was created by a scientist at MIT named Rudolf Janisch. Other firsts include, in 1982, the first genetically modified drug, in 1994, Monsanto's Flavor Saver tomato, the first GMO food, and in 2002, the discovery of CRISPR's use for human gene editing. Part 3. CRISPR-Cas9 is an advanced genome editing tool that uses a protein called Cas9, which acts as genetic scissors in order to make cuts in the DNA of an organism. Naturally, it is found as a self-defense mechanism of bacteria. When viruses attack, they can steal a piece of viral DNA and implant it into their CRISPR systems, so that the next time the virus attacks, the bacteria can recognize its DNA and use the Cas9 protein to find and remove the viral DNA from itself. In 1987, Yoshizumi Ishino was the first to describe what would later be known as the CRISPR mechanism in E. coli bacteria. In 1993, Francisco Mojica discovered a similar version of CRISPR in other microorganisms. Through research, scientists were able to determine that the CRISPR mechanism consisted of numerous base pair repeats, separated by spacers, which were discovered to be bits of viral DNA that were taken from them after attack. These spacers were what allowed the bacteria to recognize the virus the next time they were attacked. After this discovery, the scientists realized that the CRISPR mechanism could be used to edit the genome of any organism, making it the ultimate tool of gene editing. Part 4. The most important part of the CRISPR system is the protein Cas9. This protein is an enzyme that functions like an endonuclease, having the ability to cut DNA strands at specific locations. The things that tells it what to cut is called a piece of guide RNA. When a virus attacks a bacterium that has its viral DNA in its CRISPR sequence, the bacterium recognizes this and starts to initiate transcription of its CRISPR DNA sequence into a piece of guide RNA. The guide RNA then binds with the Cas9 protein and acts as a finder, directing the Cas9 enzyme to any piece of DNA within the cell that matches it. When the enzyme finds a matching piece of DNA, it then makes a cut, resulting in the viral DNA being removed. In gene editing, the CRISPR-Cas9 system can be injected into an organism with a specific piece of guide RNA to cut the organism's DNA at any spots that the researcher desires. After the cut, DNA can even be removed or inserted into the cut area, allowing for even more genetic possibilities. Part 5. One of CRISPR's primary uses is in disease control. An example is HCM, a heart disease caused by a genetic mutation. In 2017, researchers injected the CRISPR system into 54 embryos that had the mutation, and as a result, after 18 hours, around 36 of them were completely free of the disease. Another use of CRISPR in disease control is creating resistance. The same year, researchers in China increased HIV resistance in mice by using CRISPR to replicate a gene that made them naturally more HIV resilient. They hope they can apply this to humans as well, by taking genes from someone with natural HIV resistance and introducing them to someone without, increasing the entire population's resistance.